Good afternoon, everyone. Welcome back to the second session of today's FDP program. Now, I would like to request our Vice Principal, Dr. S. Sridhar sir, to welcome our resource person.
So, good afternoon, everyone. It gives me immense pleasure to welcome you all, the distinguished speaker, the faculty members, researchers, and the participants to the today's last session of the FDP program, bridging the small molecule and the large molecule CMC and industry perspective. This topic is a highly relevant emerging area in the pharmaceutical sciences. Understanding the transition from the small molecule to the large molecule therapeutics is essential for the developing the next generation of the medicines and for preparing our students and researchers for the future challenges. I confidently say that this topic will give very valuable insights and encourage meaningful discussions and enhance our knowledge of the modern pharmaceutical research and development. I sincerely thank our resource person, Dr. Varaprasad Garu, for accepting this invitation, and he is going to deliver a very good session today. So, once again, I am warmly welcome everyone, and I wish you all a very informative and fruitful session. Thank you.
Thank you, sir. Now, with an immense pleasure, I would like to introduce our today's resource person, Dr. Prasad Chandrabatla garu. Sir is an accomplished analytical R&D and CMC leader with more than 20 years of experience in pharmaceutical and biopharmaceutical development. He currently serves as Associate Director of Analytical R&D at Veranova, where he supports the development and characterization of antibody drug conjugates, linkers, payloads, and other complex therapeutic modalities. His previous industrial experiences were added with Penn Life Sciences, Sanova, Analytical USA, Aragon Life Sciences India, and Dr. Reddy's Laboratory. His expertise includes the analytical method development, validation, technology transfer, advanced mass spectroscopy, quality systems, regulatory compliance, and analytical life cycle management. He has extensive experience supporting the regulatory submissions, global health authority inspections, laboratory capability development, and cross-functional CMC programs. Dr. Chandrabatla is also passionate about mentoring the scientists, sharing the industry knowledge, and bridging the academic education with current pharmaceutical development and regulatory practices. I welcome you, sir, for this session. Over to you.
Thank you very much for the great introduction, and thank you for Sridhar sir for giving this opportunity to give a talk in the faculty development program. And I thank the Malaradi College of Pharmacy principal and the administration to create such a great opportunity. So today I would like to discuss about how modern pharmaceutical development increasingly requires us to bridge what we traditionally call small molecule and large molecule sciences. So the topic which I choose is bridging small molecule and large molecule CMC, right? And here I'm giving and taking an example of antibody drug conjugates, which is the currently uh, trend in the market. The all the market everywhere, if you see globally, even if you take USA, China, even India, now this is the emerging field, and everyone is focusing in the antibody drug conjugates. So let us go into the topic and understand how does it helps us. So if you take traditionally pharmaceutical sciences, we often organize fairly into different areas. Why the modern pharmaceutical development has changed, if you see, like we have medicinal chemistry, we have pharmaceutics, we have analytical, we have pharmacology, we have biotechnology, we have quality and regulatory affairs. So these all are we study in the academics, and the same different department areas are existing in the industry. So what are the traditional drugs? If you take the traditional drugs, it is a one molecule, a small molecule, or a biologic. So, like for example, if you take a paracetamol, we used to have a single drug. Eventually, the trend moved that instead of taking 3 to 4 tablets, we got a fixed dose combinations, having a multiple drugs in a single tablet, so that the patient will be easily take one tablet, which cures a couple of uh, diseases. And what is the today's complex therapies? If you go to the today's complex therapies, there is biology, chemistry, manufacturing, analytical sciences, and regulatories, right? So these all are required to address the today's complex therapeutics, and the structure is still useful for teaching fundamentals, but model therapeutic products increasingly cross the boundaries. So the important shift is from working in isolated scientific silos to working as an integrated development team. So if you see, when a drug is coming into the market, so chemistry, they manufacture, biology, they process, and analytical sciences test, and the regulatory science help us to submit to the regulatory. So these all are should work together. These all departments should work together to release a drug into the market. Here, for students, this means knowing an individual subject remains essential, but also need to understand how the subject contributes to overall product development decision. So ADC. So ADC is a one product with the three different sciences. So first we have an antibody where the biological targeting component, it recognizes a specific antigen and directs the therapy towards the intended cell. So if you take the ADC, there are three components here. One, the center one is the antibody. Let me try if I can access the pointer. So sorry. So the center one is the antibody, the blue one, and there is the yellow one here. It is called linker, and this is a payload. So antibody is a large molecule which is a biologic, and this is the linker which bridges the payload and the antibody. So payload is nothing but the cytotoxic drug, which is a medicine. So each of these compounds can be studied separately, but the patient receives complex ADC. Therefore, development cannot treat antibody, linker, payload as a three unrelated items. So that is why ADC is such useful teaching example for connecting biology, medicinal chemistry, analytical chemistry, pharmacology, quality, and regulatory science. So what is the architecture of antibody drug conjugate? As said, the antibody drug conjugate has consists of three main compounds. One is antibody, which provides specificity. Its role is to bind disease-associated antigen, typically on the surface of target cell. Am I going too fast? Please let me know, sir. Okay. So the linker connects the antibody to the small molecule payload. If you see here, this linker connects antibody to the small molecule payload, and the linker has to provide right balance between stability and release. If this linker is unstable, the payload may release faster, so it attaches the wrong side. If it is highly stable than required, the drug release will not happen at the appropriate time. So and the payload is the cytotoxic active provides a cytotoxic activity after the delivery. From CMC perspective, CMC means chemistry, manufacturing, and controls. We all know. However, we do not stop describing the structure. We ask whether the product has correct identity, whether expected purity profile, whether it maintains the biological potency, and whether it remains stable throughout the shelf life. So even the simple architecture immediately creates multiple analytical questions. So as we know, like if this is a it's similar like a fixed dose combination, but having biological and small molecule combination. That is the antibody drug conjugate. So if like explain, if it is the linker is unstable and the payload is not released and the antibody is degrading before or it is not going to proper antigen, so there is no use of taking this medicine. Instead of no use, it may give us adverse effects. So here next is we'll go a little bit elaborative ADC architecture component by component control. This slide takes the same architecture like we discussed before. Here, the antibody determines antigen specific and contributes much more structural complexity. And the linker determines attachment chemistry, plasma stability, release behavior. The payload delivers cytotoxic effect. So if each component can introduce different impurities, degradation pathways, therefore we need both component level control and conjugate level control. For example, we may evaluate if we take antibody, we should here we should treat as a three individual compounds or also like a single compound. We should see the drug to antibody ratio, means what is the drug to antibody ratio, how many drugs, how many payloads are attached for an antibody. I can explain more why different number of drugs will attach to the antibody in the coming slides. And we should also monitor free payload, free linker, any aggregation happening, any fragments are happening, or any charge variants we see. Right, these all are important questions to make sure that the drug is effectively delivered to the disease site. So based on this, what we understand is no single method can answer all these questions. There are different requirements, critical quality attributes, which impact the drug efficacy. And so the one's analytical method will not answer all these questions. So the main message is ADC quality depends on coordinated control of antibody, linker, payload, not testing any one component in isolation. Next slide. So what is the mechanism of ADC from binding to cell death? If you take ADC, the first step is antigen binding. So the antigen and antibody binds together, and the antibody portion recognize the cell surface target. If that binding is compromised, the rest of mechanism is already risk. As we see, like antigen, if the antibody is not bonded to the right antigen and it targets some other cell, it creates other side effects and risk as well, right? Because that is the beauty of antibody drug conjugates. The antibody is designed in such a way so that it reaches the clear antigen where it has the cancer cell. The ADC, sorry if I mispronounced, ADC is mainly for the cancer treatment. So here the mechanism is the antibody attached to the antigen, and after binding, the ADC antigen complex internalized through mediated endocytosis. So it goes to the endosome, it mediates to endocytosis, and then endosomal and lysosomal compartments. So after it reaches the endosomal lysosomal compartments, where the ADC is exposed to the enzymatic conditions. Then, depending upon the linker design and conjugation stability, it the linker cleavage will happen and the antibody degradation also will happen. So we don't need antibody. The antibody is just the transfer vehicle until it reaches the correct antigen where the disease cell is there, right? And once it reaches this enzymatic degradation, the linker cleavage will happen. The linker will be cleaved from antibody, and also antibody will degrade. So antibody, if it degrades in the body, there is no harm at all to the any cell or any part of the body. Then the linker cleavage happens, and the drug will release to the target binding site. So here, when the payload is attached to the target binding site, the released payload engages an intracellular target and ultimately produces a cytotoxic effect. So it kills the cancer cell. Now, this means what we should do as analytically. So when to confirm that the binding is proper, we should perform a binding assay to recognize whether target recognition is maintained, and does the binding assay alone demonstrate the entire mechanism? The answer is no. A cell-based potency assay can integrate several functional steps, including binding, internalization, release, and cytotoxic activity. Then does it answer all the testing, and does it make sure the drug is reached to the proper disease cell? No. Then we should also have a stability indicating method which helps us to identify premature release. So what does the stability indicating method does? It ensures the stability of the linker, it ensures the stability of payload. What if, for example, if the linker has less stability, it is not stable until the it degrades in the right place, and there is a scope of the cytotoxic drug released in between and may attack the healthy cell instead of the diseased cell. So, which creates a big side effect because the high potent, high cytotoxic drug will be attached as a payload. That is the reason we should also perform the cell-based assay, and that is why a potency of ADC is system property. It's not simple potency or a free load. So the complete conjugation has no function through a sequence of connected biological events. So this mechanism is viewed also important for comparability that the process, site, scale, and any method changes. So what are the different conjugate chemistries? Like I told in the previous slides, the drug to antibody ratio, right? So there are three types of different conjugation chemistries. Now, the industry, there are a lot of different conjugation techniques they are developing. One is lysine conjugation, two is cysteine conjugation, and three is a site-specific conjugation. We're coming to the lysine conjugation, the amide bond formation with lysine amines. So it forms NHS esters, malamides, etc. So what happens here? There will be high drug loading for the key features. At least 40 to 60 lysines are there, and random conjugation will happen. For example, in the market now, Kadcyla, it is used for the breast cancer. So here the point is when we do lysine conjugation, it creates broad range of position isomers because of multiple lysine residues may be available. Then they developed the cysteine conjugation. So what is the advantage of cysteine conjugation is it is more controlled than the lysine. So the drug to antibody ratio will be approximately around 4. So it provides more predictable loading patterns, but the product can still contain multiple drug to antibody ratios. So example, Adcetris is already in the market, brentuximab vedotin. Then it gives us some sort of site-specific approaches are designed. Next step is after cysteine conjugation. Okay, then the researchers found it is giving some controlled drug to antibody ratios. You know what is drug to antibody ratios? The number of payloads or the number of cytotoxic drugs attached to the antibody. So the researchers found to control more because if you need only two cytotoxic drugs, having more potent drug also may damage the cell, right? So we only need what is required to our body to fight with the disease. Then they came with a site-specific approach, which is also called engineered conjugation, site-specific conjugation or engineered cysteine conjugation. Here, this is designed to improve control over where how many payload molecules are attached. So they modify the antibody in such a way that there are only limited open thiols so that the the expected payloads only can attach to there. So here the conjugation chemistry is not only a synthetic choice, it determines what kind of product variants we expect to see and therefore what analytical strategy we need. So next is conjugation chemistry is the main source of ADC heterogeneity. So what we learned before is like with lysine conjugation, we may see broad positional heterogeneity. You know, right? Because we already discussed there are multiple sites so that multiple payloads may attach. So we see a broad positional heterogeneity. When coming to the cysteine conjugation, the drug to antibody ratio DAR distribution may be more predictable, but we still see we need to control some individual species. So we know, like we have only two cysteine sites or four cysteine sites. We can predict there will be controlled drug to antibody ratios. But when coming to the engineered cysteine or site specific approaches, it can improve batch consistency, but they still require confirmation of site occupancy and product distribution. So conjugation site occupancy includes like stability, PK, potency, and safety. So what are the analytical techniques we use? We use HIC, which is hydrophobic chromatography, and we use LCMS, we use UV visible. Here, if you take again in the three techniques, HIC is the column we use for HPLC, and LCMS, and if you compare with three, doctor antibody ratio, when we test an ADC under UV visible, we know that UV visible just works with the Beer-Lambert law, and if we take 280 nanometers for any protein, we take 280 nanometers, right? So if you take any protein under 280 nanometers, it won't detect only the antibody or the drug. It detects any other impurities which are active at the 280 nanometer UV visible range, right? So the method is not so precise, and we can't say the get make sure we can't confirm the value is appropriate because there any compound in the ADC we have antibody, drug, linker, and any impurities generated due to the manufacturing of linker, any impurities generated during manufacture of payload, or any modifications generated during the manufacture of antibody. So there are any all these type of components or the chemicals at the 280 nanometer can be recorded. So that is the reason we can't blindly go with the UV method. Okay, what is the next best method? Choose HPLC. HPLC is much more precise than UV, but it again comes to the similarity to the UV. It is precise because we do the chromatographic separations and do the integration. For example, if we get 3 to 4 peaks at a similar or retention time, how do we make sure which peak is really uh, a drug to antibody ratio, which peak is an unconjugated linker, which peak is unconjugated payload, right? We don't know. We get a multiple peaks. Some are more intense, some are less intense. We may think high intense peak is a ADC, and we may think less intense is the ADC. So we can't predict the data. So for that reason, what we use LC mass. So LC mass is the precise instrument, the next version to see to give the peak assignments. So what the people do industry for the LC because LCM is more expensive. All the quality control laboratories will not use the LCMS. So in the R&D stage itself, they characterize the ADC, they run the HPLC through LCMS, and they assign the peaks. So based upon the mass of the peak, they assign which peak is what. It is ADC or unconjugated finger because every compound will have its own mass, right? So they assign the um, peaks and then transfer the same method to the HPLC for the QC release. So now we have the same HPLC, we have the same column, same instrument, and run. So what is the difference if we run HPLC method alone and the LCMS? When the LCMS has its own limitations that we can't use high salt buffers. We can use the like 0.1% formic acid or 10 millimolar ammonium acetate or max is 10 millimolar or 20 millimolar ammonium format based on the instrument manufacturer recommendations. So that is the reason we should develop an help CMS compatible HPLC method so that when we transfer the methods again from LCMS to HPLC for quality control laboratory, they use the same conditions, so the retention time will not change. We already know the peak assignment, which retention time or which RRT we get the what peak, and we can assess the drug to antibody ratio there. And there are different other techniques like SEC, size exclusion chromatography, or capillary electrophoresis methods, which can monitor aggregates, fragments, and other products. During comparability, we were not only asking whether average drug to antibody ratio is similar, we may ask whether the DAR distribution, just remember I'm saying DAR means drug to antibody ratio, anywhere I'm saying in next slides, conjugate site distribution and impurity profile as shifted. So here the takeaway is conjugation device uh, design drives the analytical requirements. So here we discussed till now about the ADC characterization and the analytical requirements. But coming to here, there is one more thing here called small molecule, which is called the payload or linker. So the payload is a small molecule and linker on the is also a small molecule. Here I want to give a small introduction that uh, till now we are talking only the ADC because I took this as an example. So antibody versus small molecule conjugation where it started, it's widely spread now. Now the industry is also moving to the different type of conjugations. When the researchers understood we can conjugate antibody and drug and create good kind of medicines for the cancer treatment. Now they started conjugating everything. Now the industry is looking towards antibody oligonucleotide conjugation. Both are large molecules, and the because antibody is only to drive it and make it to correct to the proper antigen, whereas the oligonucleotide is the cytotoxic nuclear, but it is less cytotoxic compared to the small molecule payload. So small molecule payload will be, for example, will be MMAE. And oligonucleotide is less potent but more effective. So they started working on conjugating antibody and oligonucleotide, and also they started conjugating antibody and peptide. So this is the current trend in the industry going, and this may be future. We don't know. Like cell gene therapy is also there parallelly. And coming to the small molecule, this payload brings us more traditional small molecule CMC framework, right? So this is the existing, or why I said traditional means we already have this all established throughout the industry of the CMC manufacturing controls for the small molecule. All the industry has a good experience here. So here we are concerned with the chemical identity, assay, purity, organic impurities, stereochemical control where relevant, solubility, any residual solvents used there, and chemical stability, elemental impurity. So when we take the drug, all these things go into our body. So these are to be controlled. For example, there are different classes of residual solvents we use. There are different types of chemicals we use. Because when we manufacture a payload, again, it has its own intermediates. So the payload will not be manufactured as is in the first step, right? There are multiple steps there. They take raw materials, they take chemicals, and there will be multiple steps of for the intermediates. At they all perform chemical reaction together to get the final product. During all these steps, there will be different types of impurities. There will be process impurity. There will be stability related impurities and also there will be side effects of the drug. So these all should be checked, like nitrosamines. That is also one of the FDA requirement. We need to verify all these parameters before the payload is even released to attach for an antibody. So here the exact analytical methods will depends on the chemistry, but the overall mindset is familiar. Define the chemical structure, understand the impurity profile, understand the degradation pathways, and establish appropriate controls. So how do we understand the degradation pathways? So okay, we know the process chemistry. The manufacturing person or the R&D person, when he is designing this molecule, while the process is undergoing, he will say, hey, during this process, during some XYZ reaction, this impurity may generate. Then in the toxicology states, I don't want to go again a complete different subject, but there is a different phases of just I want to give a brief. There are different phases of drug development, like toxicology, clinical, phase one, phase two, phase three. During the toxicology stage itself, they say if any impurities are generated in this molecule are toxic or not. Are there carcinogenic impurities, mutagenic impurities, or any unspecified impurities? Are they affecting to the body? So these all how so will be generated in different steps, like we discussed process chemistry. Well, the process there are some impurities generated. Coming to what are the other scope of stabilities, we don't know what kind of impurities will generate when we put the drug in storage, right? When we buy a paracetamol, are we taking immediately after manufacturing? No. We store that medicine in for 1 year, 2 years, we get expiry date. So for that, they put in stability at different temperatures, and they put the different force degradation studies, and they see how much it's degrading, and if it degrades, what kind of impurities are generating, and that all to be characterized before it released into the market, or if before the payload is allowed to conjugate with a drug. This all to be uh, assessed. So here, as I said, the important point is in the ADC, the payload quality has to be understood before conjugation, right? And once the payload is attached to large heterogeneous biologic, analytical interpretation can become more complicated. So, small molecule characteristic remains fundamental part of ADC development. Next, coming to the large molecule CMC, the antibody. Here also again I want to reiterate the same, like we discussed for small molecule, there will be chemicals, there will be intermediates joined together to prepare a payload. When coming to the ADC, either it is considered as three intermediates. Antibody is one intermediate, linker is one intermediate, and payload is one intermediate. So once these all are conjugated, then it is called a finished product, right? So when we take the large molecule CMC, here the antibody, we are not interested in only in the molecular identity. We also consider primary structure, what is the second order structure, what is the higher order structure, what is glycosylation, is there any change variants happening, is there any aggregations, are there any fragments, binding activity, is there any biological potency and stability. So antibodies, it has its own characteristics, which all of these might happen during the due course before it is conjugated or after conjugation. Since the biologic can change in multiple ways, orthogonal physiochemical and functional methods are needed here. So here, if you see, there are different techniques like LCMS, which checks the intact peptide, SECMALS, CSDS, CIEF, HIC, SPR, ELISA, cell-based assays. So these are all different techniques to see the key attributes. So the main message is that even the antibody also should be well characterized like payload before it is conjugated. And if the starting antibody contains structural or functional variability, that variability can carry forward the conjugate product. So if, for example, we are telling that we are verifying the drug to antibody ratio. If we run the sample as is in LCMS and predict the data, we can't get it. If there are any modifications in the, because as we discussed, right, there are some engineered conjugations. So they deliberately modify the sequence of antibody so that it has a control number of cysteines activated to conjugate the drug. So unless we know that modification, we exactly give that sequence in the mass spec, we can't get the exact drug to antibody ratio. If we give the wrong sequence, it gives the wrong value. So that is the reason we should know all these parameters before going there, and it should be well characterized. So what we understand now at this point, we have two analytical traditions: small molecule chemical control and large molecule structural control. and functional characterization. So characterizing after ADC is formed. So these are all coming to as a single product. So okay, now we discussed all these parameters. So why analytical sciences matter and what is the critical quality attributes? So critical quality attribute is nothing but which parameter or which quality of the drug is impacting the identity, because we should know which identity, right? For example, if we call some person, they have this identity. Same way, we should have an identity for the drug we take first. So and purity is the purity of the protein or a payload, how much pure it is. More impure gives, we don't know what kind of impurity impact us. Potency, safety, stability, drug to antibody ratio, aggregation, and change variant. These are different parameters. So here the important point is not to create the longest possible list. The important point is understand which attributes can meaningfully after product quality, safety, performance, and therefore require appropriate monitoring and control. Someone may say, you are creating number of tests. Why do we need all those things? But every test has its own significance. That is the reason it is called as a critical quality attribute. So any of these parameters is not assessed properly will set will surely impact the performance of the drug, maybe adversely or not actively. That is the reason the analytical sciences translate those quality attributes into the measurable evidence. So the analytical sciences is a decision support function for the formulator. So we manufacture a good drug, we manufacture a good payload, manufacture a good antibody drug conjugate, we designed a good antibody, right? How do we know? So we need some decision making support. Sometimes the process department manufacture this ADC or any drug and submit to the analytical, and when it is failing, they come and sit with analytical, understand which data is impacting. So which quality, critical quality bit is impacted. Based on that, they do call reverse engineering, or they understand where is the problem and fix it. So the analytical department will we do generate data, but the purpose of data is to answer questions. What will be the questions from regulator? Is the product what we expect, which is identity, right? We are saying it's a paracetamol. How do we know its identity? To ensure the chemical structure with a different test characterization. Is it sufficiently pure? Is it structurally, functionally consistent? Has it changed during manufacturing or storage, which is called a stabilities? Is a process change comparable? Because even we have proper process design, sometimes it may variable, the conditions may be variable, temperature, the flow, speed of the flow, anything may impact the process. So that all should be analyzed. And as development advances, this data supports method selection, specification, stability programs, comparability, and regulatory sufficiency. So I think we all know about what is specification. Specification is nothing but a set of boundary we put for each parameter. For example, if you take assay of a drug, if we say it's 98 to 102% for drug substance, and for finished product we give 90 to 110. So these are the predefined specifications. So that is the reason the CQA as provide as a bridge between product science and control strategy. So here if you see, my heading is industry does not test methods, industry answers questions. Here, why do we teach analytical chemistry? It is natural to organize subject technique, HPLC, mass, electrophoresis, spectroscopy, and so on. If you see, why do we learning the technique itself is not sufficient. In product development, we need to have the stronger starting point of scientific question. For example, if you ask what is the molecular identity and comparison of the product, then what LCMS will answer that. What is aggregation and molecular size distribution? A SECMAS instrument will answer. SECMAS is nothing but a size experiment chromatography with laser distortion. And if a question asks what does the drug to antibody ratio or hydrophobic distribution look like? The Hick method technique will answer that question. If someone asks, what is the question about charge heterogeneity? What is the charge impurities profile formed there? What is the charge heterogeneity there? Then we use ICIEF or ion exchange approaches. So if someone asks about the does the antibody still binds the target? Are you sure? Then the ELISA, SPR, BLI. This is one more technique to answer that question. If the question is whether the complete ADC produces the intended biologic effect, then the cell-based potency assay. So we give ADC to the human body. How do we know it entered into the body and attached to the proper antigen, and the cytotoxic payload is released properly, and it's effectively killing the disease cell. For that, we do the cell-based assays. So this way, the of thinking is a powerful education instead of asking students what does the instrument measure. We can also ask what product quality question would make you to choose this instrument. What are the limitations? What are the additional method do you need? For example, I like in the previous slide, explained about the comparison between UV instrument, HPLC, and LCMS. So just don't learn the instruments. Even now, in industry of some people having experience, they just follow what is there in the method and they just run the instrument. It doesn't make any sense. If there is really a problem arises, you should know what the instrument is used for, how does it give the data. So that is always important. So what are the orthogonal analytical characterization of ADCs? So orthogonality means here we use complementary analytical techniques to examine the product from different scientific perspectives. For ADC, we may evaluate mass and structural identity. We may evaluate molecular size and aggregation. We evaluate hydrophobicity and dark distribution, and we charge heterogeneity, binding activity, and biological potency. So these are all different dimensions of product quality. One method may highly sensitive to particular change or relatively insensitive to other. If we take one method or one technique, will not answer all these questions. So what is the example? If you want more deep to understand, a chromatographic method, if we take, it will just reveal the impurity peak, but it can't say the structural identity. Like we discussed, the HPLC alone will not give the identity of the peak. It gives we may separate the chromatography and get multiple impurity peaks, but we don't know what peak is what until it is confirmed or assigned by LCMS, right? And if you take physicochemical method, it may show structural similarity, but the functional assay is still needed to demonstrate the biological activity. So here for the faculty, this another useful teaching point is ask students why two methods might be needed to answer what appears to be one quality question. So we are doing 10 number of techniques, 20 number of techniques. So what is the significance of each technique is always important to learn. If you see this orthogonality toolbox, which I made like a flow chart, right? If you see the orthogonality means I organize the analytical toolbox by type of question being answered. For example, HRMS, it can support, let me point out. If you take LCMS like HRMS, it can support intact mass, subunit analysis, dark species, and post-transfer modification. If you take electrophoretic methods such as HPLC and chromatography, SEC, CESDS. So here I arrange this flowchart like orthogonality, what each kind of instrument can do, multiple techniques. If you see, all the techniques are interrelated. all the techniques are interrelated. One instrument give multiple characterizations, and all are synced here. All are interrelated, connected. Means these all tests are performed for a single moiety called antibody drug conjugate. So, what is the analytical CSC? That is use complementary methods to reduce method-specific blind spots and align each method with one more CQA and acceptance criteria, define stability indicating capability during method development, build the final release and characterization panel from risk-based evidence. For example, if you take what is defined stability indicating capability during method development, if we create a chromatographic method, right, for HPLC impurity method, if it is giving 2 or 3 peaks or 10 peaks, doesn't mean that it separate all the impurity peaks there. There will be co-eluting peaks or a couple of impurities merged into one chromatography. That is the reason stability indicator we do forced degradation. We check by hydrolysis, acid, light exposure, all different techniques to make sure how can impurity form. If in what condition, every condition there is sort of impurity will form, right? So we make sure that we check all impurities are detected properly in the method. That is the reason that's called a stability indicating method. Next is integrated characterization. So now we discuss the antibody separately, payload separately, payload and linker both are small molecules. So now we are going for a integrated characterization of ADC, which covers the ADC monoclonal antibody, drug, and linker. So here, if you see the slide, illustrates the end-to-end analytical control. For complete ADC testing may include intact subunit mass, drug to antibody ratio. We need to the peptide mapping to make sure that the proper peptide mapping also confirms the site conjugation and glycosylation. If any glycosylation pattern is happening, what is the purity and potency? For starting antibody, we evaluate identity, glycosylation, purity, and higher order structure. For the payload, we appropriately identify the chemical purity, identity, strength, assay, purity, and impurity control. Similarly to the linker as well, right? So the control strategy connects these materials rather than treating each specification as an independent table. For example, if you take a free drug and free linker, can be important impurities in the final conjugate. So any process changes may alter drug to antibody ratio distribution or aggregation. A slight change in the process because the antibody we know can impact well in small variation. So those all are to be verified. And if a site or scale change may also require comparability evidence. So if you take analytical method change may also require to demonstrate the product quality conclusions remain consistent. Ultimately, these all information will support to the CTD module 3, which is the regulatory submission document, which we need to which covers all materials, process, drug substance, drug product, specifications, stability, and comparability. In the CTD 3, module 3, which is used to submit this drug product, we need to give where we procure the reagents, what are the COS of that, we need to provide the intermediates details, we need to provide the finished product details, what are the analytical methods we use, what are the validations we perform, what is the calculation what is the different techniques we performed, are they calibrated, all the details should be submitted. So ADC CMC success requires end-to-end control from component to the final conjugate. Stability of ADC's key considerations. So how do we ensure the stability? What is the main points in the stability? ADC stability is challenging because we have to consider both physical and chemical degradation. Physical changes include aggregation, fragmentation, and particle formations, whereas the chemical changes can include linker instability, payload degradation, deconjugation, oxidation, and other modifications. Like we discussed, we there are different parameters we do. If the linker is not stable, then the payload may release before it reaches the proper cell disease cell site. And if the linker cleaves, the payload will go and eat a healthy cell and creates further damage instead of curing the disease. Deconjugation, similar thing happens. Oxidation. So we do characterize different types of uh, parameters. Is there an impact of oxidation? Is there an impact of deamidation? All these parameters we do in the characterization of antibody. So these changes can influence by pH, in the body pH also, right? Temperature, any excipients, if the drug is exposed to any light exposure, freeze-thaw conditions. What is why freeze-thaw means? Most of the antibodies, ADCs are stored in -80 or -20 degrees. So when it is sold or when we use, there will be multiple times of we take out from the temperature, put on the room temperature, use it and put it back, right? Or shipment. When they put in -80, when they ship, if they may put in dry ice, but there's a time in between this both transportation. So that's the reason multiple freeze thaw. How many times we can freeze and how many times thaw, how much time it can be stable on the room temperature. These all assessments to be should be performed like a freeze thaw conditions and any other storage or handling conditions. So, for example, if you go to any medical shop, the medicines will be stored, right? So, what conditions it be stored? We all have all this in packing insert. What conditions are in the it should be stored, like light sensitive, exposure to light, room temperature, put in refrigerator. All this all comes under these studies. So, a good stability program therefore needs to method to capable of predicting meaningful changes in the relevant sequence. So, for complex products, stability is best understood as profile across multiple attributes rather than one assay result. So, even again, the stabilities method, we should check all these parameters. Just if you take the assay of the product, doesn't mean, for example, if we get 95% of assay, what happened to the next of 5% in 100? Are there any carcinogenic or mutagenic impurities in the list of 5%? Or any other side effects will give you the impurities? So, how do we know? Unless we characterize that impurities, right? So, all these methods to be evolved. That's the reason post degradation stress studies can valuable during the development, and it understand the help us to understand the degradation pathways and determine analytical methods. So, what is the analytical journey of ADC here? So analytical strategy is not static here. In discovery and early development, the emphasis of is often on rapid product understanding, identifying major risks, and the establishment of fit for purpose methods. There are different phases, as I said, phase one, two, three, right? So in early phases, if we perform complete validation of the method, doesn't make sense because when the process is changed, we need to validate the method again. That's the reason early phase we do fit for purpose methods. Means the method should be fit for what is intended to use for impurity identification. So when the phases are different phases, we move on. The manufacturing techniques may be changed. Hence, the complete analytical method validation or methods are important in the phase three. Phase one, phase two also we have methods. Even the regulator gave a guidance recently. No need of deliberate stability data, but good to have in the phase one, like that, right? So as the program matures, the methods become robust, and we also clearly understand what is the critical quality of attribute because we learn from the failures, right? So when the program is moving forward, we understand more critical quality attributes, more degradation pathways, and then the specification begin to the mature. And when it comes to the commercialization, the analytical strategy supports release, stability, comparability, continued process verification, investigation, and life cycle changes. So a strong early analytical strategy creates knowledge that makes later development more efficient. So if with the analytical technique, if we know why the product is failing, what is the critical quality attribute, where can we see what change is impacting this? For example, there is a nitrosamines. Nitrosamines If we ask a tertiary or binary amends, right? Then if their process is about 70 degrees, they do a simulation study and submit sample to analytical. There they see really it's generating impurity or it is safe, not generating impurity. So always the analytical strategy creates a good knowledge and make later development more efficient. How ADCs connect core pharmaceutical sciences. So we discussed, right, in the first slide itself, in pharmaceutical sciences, there are different departments, like different areas. We have medicinal chemistry, we have pharmacology, biotechnology, pharmaceutical analysis, quality, regulatory affairs, right? In the academic curriculum, these subjects may appear different courses, but even different years of studies. In industry development program, the same product requires all teams to interact. Some are specialized in pharmacology, some is specialized in medicinal chemistry, some in biotech, some in pharmacology, right? So this creates useful education. For example, if you take ADC, you can see all things. The medicinal chemistry will be helpful in the payload design. The biotechnology helpful through antibody structure and function. Pharmaceutics through formulation and stability. And analytical sciences through critical quality attributes and method selection. And pharmacology knowledge helps through mechanism of action. And regulatory science through control strategy. So the value of ADC exam not only that is a modern modality, it helps that students to connect knowledge that is otherwise taught separately. So we think why we do medicinal chemistry, what we'll do in future, or some as biotechnology. When I am studying, people used to say, "Oh, biotechnology is the future. Chemistry is nothing, or pharmacy is nothing." So we are in a confused stage. What? But when it really comes to the industry, every department, every subject we learn is important. We should move with what opportunity we get, and we move on from there and develop the skills. So, what are the common analytical mistakes in early development we do? One common mistake is relying too heavily on a single analytical method. So, we discuss all these slides. We just rely on single analytical method. I think I'm running too much time. I'll finish the slides quickly. So, if you develop a single analytical method, for example, if we take SM method and we rely the product is clear, we know that it is pure, doesn't matter. And second thing mistake we do is developing methods without clearly connecting with them to critical quality attribute. So developing some XYZ method, right? We ignore the critical quality attribute doesn't make sense to release of any drug. Next is lack of orthogonal characterization. That is the biggest mistake people do. If we different methods do not cover complementary product attributes, important changes may remain undetected or poorly understood. And fourth is insufficient stability understanding. And finally, team comes sometimes try to establish final control strategy too early before sufficient product process knowledge is generated. If in early phase itself we create all this degradation and final methods, when the phase excels and the process changes, there will be a lot of different impurities or different kind of changes may happen for it. So that is again the waste of time or waste of resource. So these are not simple laboratory mistakes, these are the development stage called as a development strategy mistake. So we should always have a strategy. So that is the reason what product quality question is method intended to answer, or what evidence do we need to support the decision. So what are the lessons learned from industry? The first lesson is that quality is designed into product and process. It is not created by final testing. Second lesson is the analytical method should answer scientific questions. And third lesson is the product knowledge drives the regulatory success. Fourth is the CMC evolves through life cycle. It is not like process change, sites change, method change, specifications may evolve, and new knowledge becomes available. And for the students, this means being a strong analytical scientist requires more than operating instruments. It requires connecting data and product quality, process understanding, clinical relevance where appropriate, and a patient impact. So, what industry experts from pharmacy graduates? So, all the motto comes back to there. What is the industry expectations here? More about product understanding, just not the technical it needs technical competency, not the technical competency enough. Graduates should understand the product they are testing. They should able to interpret the data, not simply generate chromograms and numerical results. They also should have a basic understanding of both small and large molecule CMC because the trend of market is moving. In the olden days, there is specialization called learning more and more about lesser and lesser. Now, any field, not only pharma, you should be always there is saying called jack of all, master of none. So we should learn everything. We should be flexible to move your career. We should take the opportunity and move forward. And students should learn concepts of quality by design, critical quality attributes, analytical control strategy, stability, all these parameters. At the same time, industry also needs scientific curiosity, critical thinking. Just okay, I got the sample, I tested and give the data doesn't mean you need to have the curiosity. What happened to the data? What does it give to the answer to the manufacturer? What does the formulator understand from this data? All this, the curiosity should be there in learning. So the scientist may generate excellent data sheet, but the value of work increases when the scientist can explain what the data mean, what risk they identify, and what decision they support. Because when the formulation comes to analytical and say, hey, what do you know, or what do you see from the data, we should able to understand. When we able to understand, we also know the process. We also should know the formulation. Same way, the formulator should also know the analytical skills. He can't complete real. So we should always have the knowledge of everything. So what is the message emphasis is industry is looking for scientists who understand the product, not only the analytical method. So coming to the curriculum-oriented faculty development, because it's a faculty development program. When I talk about curriculum-oriented faculty development, I'm not suggesting the foundation subjects should be replaced. Strong foundation in chemistry, pharmaceutical analysis, pharmacology, biotechnology, and quality remain essentials. So all these areas are essential when it comes to the industry. When we get opportunity, we should dig into that deeper, and enhance our skills. So the faculty development also can introduce industry concepts such as CQS, what is critical attributes, control strategies, phase appropriate development, etc. Last week when we are installing an high resolution mass spec in our company, the service engineer came and told, sorry, I have some schedule, it's very due install. The engineer told that she installed a QTOF instrument in a university. I felt very happy. I told really I'm glad to know that universities are installing QTOF instruments. So most of the methods for the early phase studies come from the universities. Since I'm working in CDMO, we work with the contract development different companies globally. So most of the early phase, the formulations come from the universities. They are not the industries won't spend money on certain researchers until they are confident because it's a business. But universities do the different formulations, even the ADC concepts, any concepts, they evaluate starting from the universities. So the research mindset, the opportunities start there itself. So implementation roadmap, expect impact. A practical program can begin with a need assessments and a curriculum. So first identify where curriculum already addresses modern development concepts and where the opportunities for stronger integration is happening. And next is targeted faculty development activities. Like they can include case studies, short industry modules, interdisciplinary teaching sessions, guest lectures, updated laboratory discussions. So interact from the industry from the academy itself. Give the guest lectures, take to the site visits and all. Third, deliver the training in the way faculty can translate directly into teaching and evaluate the outcome. Are the faculty finding the material useful? Are the students engaging differently? Are they better? So, like, they should not just follow the books. Follow the books and interpret differently. Create the curiosity from the graduate states itself. So that's the reason the faculty development is not a one-time event. It's always ongoing mechanism for keeping education connected to an evolving pharmaceutical landscape. So what is the key take-home messages? So my first take-home message is teach the science behind the method, Students should understand not only how the instrument works, but also product quality question it answers. Second is integrate small molecule and large molecule concepts because the future of pharmaceutical development increasingly requires both. We don't know where we excel. We don't know where we get the opportunity. Simple example, I'm not saying. I did my MS in organic chemistry. I got my first job in bioanalytical. Everyone laughed who are there, they're pharmacists. They say, pharmacy background. Do you know ADMG? Do you know dissolution? I don't know. Then I went to one of the friend there. I used to go in the evening to their home in the weekends, get the books, read, understand, and establish it there. After that, I moved to CMC analytical. People said, you have 14 years of experience. Why you over to CMC? You will fail. Let me take a challenge because I want to excel there. Then I went there. I took 6 months of time, um use of time after the hours and read some things and establish it there. And in CMC, I'm now from 8 years, and now there is ADC, the new technology. They asked, "Can you take a challenge?" I took friends help like Sridhar or any other faculty people. What is the antibody? I started from there, from scratch, learn from there, and now I'm leading the ADC. So there is no defined pathway for our career or life. It's like SOP. It's not like SOP, which is pre-written. We should always capture the opportunity, develop your skills, and learn and move out from there. So when you in any time of career, if you stop learning, it means you are retired on the same day. Even like you know, even the faculty, how much changes are there from the time we studied and now. So always you should be ready to take the challenges. So for all this thing is first start studying from the graduate or school time with the science behind the anything, not just following the methods, following the literature. What is the science behind that thing? How does the drug work? That curiosity should be there, and you should also like if you are working in a bio doctor, if it any drug, if you are taking at home, start reading the literature. How does it impact? How does it release us? How does because we are all in a pharmaceutical pharmacy background, right? How does it releases? How does any impurity in it? So if you see the drug package, we throw just we take buy the thing and throw in the trash. But no, read that. Go to the literature. Create curiosity from now itself. So any questions? Sorry, I think I took more time.
రిమూవ్ చేయాలి దాన్ని మీరు చేయండి రిమూవ్ చేసాం పీపీటీకి రిమూవ్ చేయండి
Sorry, sir, if I am over the time.
No, sir. No, sir. Thank you. Thank you very much, sir. So, good evening, everyone. It was a great pleasure to have Dr. Varaprasad Chandrabatla with us for this insightful session on bridging the small molecules to large molecules CMC, especially in the analytical industry aspect. Okay, we sincerely appreciate Varaprasad Garu for sharing his valuable industry experience and practical insights into the CMC and analytical method development. This session has provided us with a better understanding of the challenges and opportunities in the pharmaceutical development from the small molecule to the complex large molecules, especially in the analytical aspect. Okay, we are grateful for his valuable time and excellent presentation. Thank you, sir. Really, when we asked you immediately, you responded on the day, and till today, you supported a lot. Okay, and your speeches really enriching our FDP with your expertise. Okay, so and next thing is regarding the faculty development program, 5-day program. As we come to conclusion of our 5-day faculty development program, in association with the APTI Telangana branch, I privileged to share a few words of gratitude. Over the 5 days and 10 insightful sessions, we had the opportunity to explore how the digital tools, artificial intelligence, and the machine learning can be effectively integrated into the pharmacy curriculum and enhance the teaching, learning, research, and as well as the academic outcomes. The excellent participation, interactive discussions, and valuable insights shared by our resource persons made each and every session truly meaningful and enriching. Okay. I would like to thank all our management, okay, and the participants and the resource person, okay, for making this 5-day FDP grand success. Okay, now I request our faculty Sridevi Garu to give the vote of thanks.
Thank you, sir. A very good afternoon to everyone. So it is my pleasure to extend a vote of thanks at the conclusion of the fifth day of the International EFDP program organized by Mallareddy College of Pharmacy in association with the APTI Telangana branch. First and foremost, I express my sincere gratitude to our respected chief patrons, Sri P.H. Mallareddy, Founder Chairman, MLA, Government of Telangana, for his vision, encouragement, and continued support in promoting the academic and professional excellence. I would also like to convey my heartfelt thanks to Chindana Krishnamohan Garu, who is the President APTI Telangana, for associating with us and giving time to making this FDP successful. We are grateful to our keynote speakers, Mr. P. Mallikarjun Rao sir from Malaysia, Dr. Ashok Tuluru sir, who is a professor and HOD from D.Y. Patil College of Pharmacy, Maharashtra. A heartfelt thanks to Dr. S. Anbuselvi ma'am, professor of Bharat Institute of Higher Education, Chennai. We sincerely thank Dr. A. Buchraju sir, who is a faculty member at Saudi Arabia. A special thanks to Dr. Rajendra Prasad from Vishnu Institute of Pharmaceutical Educational Research, Telangana, for his insightful session. We also express our sincere gratitude to Dr. Principal from Aditya Bangalore Institution. Our thanks to Mr. Tajuddin Sheikh, who is an assistant professor, MRCP, co-founder of Biodocify Hyderabad, India. We are grateful to Dr. K. N. Nurullah, who is a professor of PSG College of Pharmacy, Tamil Nadu. Our sincere appreciation goes to Professor C. S. Mujibuddin, who is the founder and CEO of Clinosol Research, Hyderabad. My sincere thanks to our esteemed convener, Dr. Sudhakar sir, who is a principal, and also extend my heartfelt appreciation to our respected co-convener, Dr. S. Sridhar sir, the vice principal of MRCP. Extend our heartfelt thanks to Dr. Chandrabatla sir, who is an associate director, Analytical R&D, Veranova Systems, Devens, US. I also extend my heartfelt appreciation to our respected coordinators Sridevi, Jyoti, and Gayatri, whose continuous efforts and coordination have contributed immensely to the smooth conduct of this FDP. A special word to thanks Mr. Tajuddin Sheikh, who is an assistant professor MRCP, for his excellent coordination and wholehearted support throughout the FDP. We are also grateful to him for being a wonderful speaker for the Biodocify session. Now, I take this opportunity to express our deepest gratitude to all our esteemed researchers who graciously shared their knowledge. Our sincere thanks to all the participants for making this faculty development program a meaningful, enriching, and memorable learning experience. Thank you all. Thank you once again. So we have come to the conclusion, and dear participants, the feedback form will be posted in the WhatsApp group. So after analyzing the attendance and the feedback given by you, we will be delivering the EFDB participation certificates by tomorrow. So thank you all for your wonderful cooperation. Have a wonderful day.